Good day, everyone. I am Dr. Adeyinka Gbadebo, your biology lecturer. Please feel free to subscribe to my channel and click the notification button. From time to time, I'll be giving you updates on topics and subtopics in biology, zoology, ecology, and genetics from time to time. So click the notification button to get updates from me from time to time. Thank you. In this lecture, I'll be elucidating the mechanism, a summarized mechanism of DNA replication. I'll be summarizing the mechanisms, the processes in DNA replication. In my previous lecture, I've been able to touch on some important components that are involved in this process of DNA replication. We treated the DNA polymerase, we treated the primer. We're also able to touch on explaining how replication takes place continuously and discontinuously on the leading strand and on the lagging strand. We're also able to mention that on the lagging strand is replicated discontinuously, forming short segments of nucleotide called Okazaki fragments. And these Okazaki fragments, they're eventually joined together. And DNA ligase eventually will join the newly synthesized DNA strands to the parental strands, forming two different strands. That is just in summary of what we have looked at in my previous lectures. In this lecture, I want to summarize the mechanisms involved in DNA replication. For simplification, the mechanisms of DNA replication can be divided into five interlocking steps. Now, what are these steps? Number one is opening up of the double helix. Number two, building a primer. Building a primer is the number two step. Number three step, assembling complementary strands. Assembling of the complementary strand is number three. Number four is removal of the primer. Removing the primer. And then the last step is number five, joining the Okazaki fragments. Joining the Okazaki fragment is the last step in DNA replication. So let's start with the first step in DNA replication. The first step is opening up of the double helix. As we know, DNA replication is actually a complex process, and as a complex process, it involves several proteins and enzymes. For this process to be functional and to be successful. And then the first step that must take place for DNA to replicate, for DNA synthesis to take place, is opening up of the double helix. Now, this first step can be divided into four different stages. Now, what are these stages? And we'll then explain the first step based on the four stages. The first stages are one, initiation, the initiation stage. The second stage is unwinding of the double helix. The fourth stage is stabilizing of the single strands of the double helix. And then the fourth stage in the first step is the relieving of the torque pressure. These are the four stages under the first step in DNA replication. Now, let me explain these four stages under the first step. So the number one stage is the initiation stage. The initiation stage actually involve some proteins. We call them initiator proteins. Initiator proteins, the moment the moment either the moment DNA is about to be replicated, the moment there is signal for DNA replication within the cell, the first stage is the first thing that happen is that there are some proteins called initiator proteins. The initiator proteins bind to a point on the DNA to be replicated, or the section of the gene to be replicated. The initiator proteins bind to a point on that gene or on that DNA. That point is called the origin of replication or replication origin. So it binds to a point, the initiator protein binds to a point called origin of replication. Origin of replication. So the moment initiator protein binds to this origin of replication, it signals the beginning. It signals the beginning of the replication process. It signals the beginning of the replication process. So the next stage is the unwinding the double helix, unwinding of the double helix, and the unwinding is usually aided or carried out by an enzyme called DNA helicase. An helicase enzyme. It is the helicase enzyme that allows the unwinding of the double helix. The helicase enzyme, DNA helicase, unwinds, from the replication origin. The helicase enzyme attached to the replication origin and starts to unwind the double helix. How does the helicase enzyme perform its action? It attaches itself to one of the strands while it then shoulder the other strands. It attaches itself to one of the strands. The helicase attaches itself to one of the strands while it shoulders the other strands and moves along the double helix that it is unwinding. It shoulders the other strands. It attaches itself to one of the strands and moves along the double helix to be unwind and shoulders the other strands and then as it moves along, it unwind or it unzip the double helix of the DNA. The third stage is stabilizing the single strand. After the unwinding takes place, single strands of the double helix will be exposed. Remember that the DNA is like a spiral staircase. It's a spiral spring, and if it has been unwind, what will normally happen for a spring, if you take a spring, if you try to unwind a spring, it will then try to rewind back. If you try to unwind a spring, it will want to rewind back. And if you are not careful in unwinding a spring, if you add too much pressure in unwinding a spring, at some point it can break off. So in this third stage of stabilizing the single strand, there is a specialized protein called single strand binding protein, SSB. Single strand binding protein. This single strand binding protein, as it is called, single strand binding protein, binds to the exposed single strands of the DNA. It binds to the exposed single strands of the DNA, preventing them from breaking, or the word is preventing them from cleavage, and then it also prevents them from rewinding back. The single strand binding protein, SSB, binds to the exposed single strands, the unwind or the exposed single strands of the double helix, preventing them from cleavage and also preventing them from rewinding. Now, the fourth stage is called relieving the torque pressure. Relieving the torque pressure generated by unwinding. That is the fourth stage. When a double helix unwind, that unwinding usually creates a torque pressure, and the reason why this torque pressure is created is because DNA is a double helix, is composed of a double helix that is like a spiral spring that is highly wound and highly coiled. So unwinding of DNA normally generates a torque pressure. And now for this torque pressure, it normally generates a torque pressure. And just imagine that unwinding usually takes place at the replication fork, and it normally makes the DNA to rotate about 1000 revolution per second. Unwinding the DNA at the replication fork usually makes the DNA double helix at the replication fork to rotate about 100 revolution per second. So the pressure is always much. The pressure generated is usually very much, and it normally generates a torque pressure. And so to relieve this torque pressure, the torque pressure is usually we want to act against. We want to make the DNA to rewind. So to relieve this torque pressure, there's another enzyme is needed. This enzyme is known as topoisomerase. And topoisomerase is also called gyrase. Topoisomerase is also called gyrase. Topoisomerase is also called gyrase enzyme. This topoisomerase is needed to relieve the torque pressure that is created by the unwinding of the DNA double helix. So topoisomerase usually binds to the double helix and prevents or relieve the torque pressure that is created as a result of the unwinding. So just in quick summary, the first step can be divided into 4 stages: initiation, unwinding, stabilizing the single strand, and then to relieving of the torque pressure that is created by the unwinding process. The next step in the mechanism of DNA replication is building a primer. And a primer, like I've explained in my previous lecture, is a short stretch of RNA nucleotides. A primer is usually a short sequence of RNA nucleotides, and usually about 10 base sequences of RNA nucleotides. A primer is needed because the DNA polymerase cannot initiate the synthesis of nucleotides. The building of nucleotide chain on the DNA double helix template cannot initiate it. So, and it is because synthesis requires a 3' end. So usually a short primer, which is a short stretch of RNA nucleotide, is usually first of all constructed. And this is usually constructed by an enzyme called RNA polymerase or primase. So the primase constructs the primer, and it is on this primer which exposes a 3' end that DNA polymerase can then continue to start building sequence of DNA nucleotides to continue the elongation process. The next step in DNA replication is the assembling of complementary strands. The assembling of complementary strands. Now, the enzyme that is needed to assemble complementary strands is the DNA polymerase 3. DNA polymerase 3 is needed for chain elongation, or what we call assembling of the complementary strands. Assembling complementary strand, which is chain elongation, takes place, follow different pattern on the two strands of a double helix. The two strands of a double helix, one strand is called the leading strand, the other strand is called the lagging strand. The DNA polymerase 3 polymerize or synthesize nucleotides, complementary nucleotides, continuously on the leading strand towards the replication fork, whereas on the lagging strand, synthesis of DNA nucleotide sequence is discontinuous. Usually forming short segments known as Okazaki fragments. So that makes the process of DNA replication a semi-discontinuous process. So the DNA polymerase usually binds to one of the strands, synthesizing it continually. We say that, remember that I said that the DNA polymerase 3 is a dimer. Dimer meaning that it has two subunits. So one of the subunits binds to one of the parent strands of the double helix. The other subunit binds to the other strand of the parent helix, and then in that way they clamp the double helix, they hold the double helix and moves along it, and as they move along it, chain elongation or DNA synthesis proceed in that order. Now, the fourth step in DNA polymerase is removing of the primer. The primer is usually removed by DNA polymerase 1. Now, DNA polymerase 1 moves along the synthesized helix, and then recognize the primer and removes it. It removes the primer, and apart from removing the primer, it also fills the gap that is created. It fills in the gap that is created with complementary bases, complementary DNA bases. It fills the gaps that is created by complementary DNA nucleotide bases. And then apart from this function that I've mentioned, it also joins the gap between the Okazaki fragments. It joins the gap. The Okazaki fragments, they are in short segments. It joins the gap. Joins the gap between the Okazaki fragments. So the DNA polymerase 1 does that. It removes the primer. It fills in the gap that is created by the primer using DNA nucleotides, and then it joins the Okazaki fragment that is being created on the lagging strands. So then, now this fifth step in DNA replication, the fifth step is the joining of the Okazaki fragments. The enzyme that is needed to join the Okazaki fragments, the continuous strands of Okazaki fragments, the enzyme DNA ligase. DNA ligase is the enzyme that is needed. The DNA ligase joins or binds the long chain of Okazaki fragment to the lagging strands. It joins it or binds it to the lagging strands, and then eventually from the parent DNA, two identical DNA double helixes will be formed, marking the end of DNA replication. Quickly, I would like to mention that the the mechanism of DNA replication that we have been explaining, that I've explained thus far, is more precisely for prokaryotes. Prokaryotes are studied in E. coli, is more precisely is more precisely for E. coli. However, there is little difference in the mechanism of DNA replication that you find in eukaryotes, in multicellular organisms, that is multicellular organisms. Okay, now what is this difference? Actually, there is similarity. In the DNA replication between prokaryotes and eukaryotes. There's a whole lot of similarities. It also involves the different enzymes and proteins that we have mentioned. The enzymes and proteins that we have mentioned in our previous explanation for prokaryotes are also involved. The DNA polymerase, the different enzymes, the helicase, the topoisomerase, the DNA ligase, all those enzymes, they are also, those steps, they are also involved. Those 5 steps that we mentioned, they are also the same steps you find in DNA replication for eukaryotic organisms. However, the little exception or the little addition to the explanation for DNA replication in eukaryotic organism is that DNA in eukaryotes is usually packaged as nucleosomes within chromosome. DNA is usually packaged as nucleosomes within chromosomes in eukaryotic organisms. They are packaged in nucleosomes within chromosomes. Nucleosomes are actually long stretch of DNA double helixes that are wrapped around histone proteins. That is nucleosomes. Nucleosomes are long stretch of DNA double helixes that are wrapped around histone proteins. Usually those histone proteins, they are like something like this. There are four units, and they are like beads. So we have long stretch of the DNA that are wrapped around them. DNA are wrapped around them like this. You have nucleosomes, and then DNA are wrapped around them like this. Each of these units is called a nucleosome. And these nucleosomes, they are packed, and they are packed in chromosomes. So that differentiates the DNA. The structure of DNA or chromosome of prokaryotes from that of eukaryotes. The DNA of eukaryotes, they are packed in nucleosomes within chromosomes. And then another thing that you need to note is that in eukaryotes, unlike prokaryotes, eukaryotic DNA usually have multiple origin of replication. You find multiple origin of replication in eukaryotic chromosomes or in eukaryotic gene. Eukaryotic genes, they are usually much more longer, or eukaryotic chromosomes, they are usually much more larger than prokaryotic chromosomes. So it usually allows for multiple origin of replication. Unlike in prokaryotes, that is bacteria, you just have one origin of replication. Also, because of this multiple origin of replication that you find in eukaryotes, you also find what we call different section or discrete section or discrete zone on a chromosome that can be replicated at the same time or at different time. on a single chromosome, there are discrete zone. On a single chromosome, there are discrete zone on that chromosome that can be replicated simultaneously or at different time. So these discrete zones on a chromosome that can be replicated is usually called replicon. It's called replicon or replication unit. It's called replicon or replication unit. It is because of this discrete zone on the chromosome or replicon or replication unit that makes the DNA of eukaryotes to have multiple origin of replication. It is because of the discrete zone. If this is a chromosome in eukaryotes, from here to here can form a discrete zone that can be replicated separately. From here to here can form another discrete zone that can be replicated separately, and so on and so forth. And each of these zones can simultaneously be replicating at the same time. So, having said that, multiple origin of replication, the advantage of multiple origin of replication is it allows for speed in replicating a chromosome. It will have, for instance, in human chromosome normally take approximately 8 hours to complete replication. Human chromosome normally take approximately 8 hours to complete its replication. Now, that is quite fast for a very long stretch of DNA that is wound up into chromosome. 8 hours is actually very fast. Now, this would not have been possible if there is just one origin of replication. If there is just one origin of replication, it will not have been possible to be that fast. But because there are many origin of replication on the same stretch of chromosome, so replication can actually continue on the same chromosome from different or discrete region or discrete zone. So everything can continue separately, can continue as discrete zone, and then eventually the whole chromosome will be replicated together, and that makes it very, very fast. So what I've just given you is the similarity and the differences between DNA replication and in eukaryotes and that of prokaryotes.